Yeah, welcome. In my talk, I want to, in principle, drive the interaction between anatomy and function from the functional side, because I have to admit that I'm originally, when I started in brain imaging more than 20 years ago, I was actually thinking, I don't need anatomy, I just want to understand the building blocks of cognition, and so I was really not appreciating neuroanatomy. And that has completely changed over the last 20 years. And the more we move now into high field imaging, which is the topic of my talk, we look at structures which are like much smaller than in the usual fMRI studies, and we need more and more anatomy to understand what we see. So I want to make that sentence at the beginning that I'm becoming kind of a convert. And what I want to show you today is a little bit a change in scope, a change in view from the perspective of functional brain imaging, namely that we want to understand better not only the macroanatomical functional divisions of the cortex, which of course to some extent, as we have heard before, linked to cytochronic borders and so on, but to really try to understand what's going on inside these areas, and can we also understand the representations and the separations within these structures. And we look at two kind of dimensions. One is the tangential one, and we look in structures which are usually called columnar structures, and the other is the depths of the cortex, and we look, as we have beautifully seen before, the laminar structure. Of course, even with high field imaging, we cannot separate in the detail we have from neuroanatomy the layers and columnar structures, but we want to make new insights into these representations by using ultra-high field imaging. And we try to kind of understand and relate these structures to anatomical regions. Important point hereby is that the understanding, even the initial steps we have done and others over the last years, are already showing that we can hopefully understand cognition better, perception better, and that is due to the mixture of anatomical and functional insights, which I want to share with you. So the first part is to give you a bit an overview of macroanatomy. So usually when we talk about macroscopic scale in functional imaging, we refer to localizing brain regions, functional brain regions, like the face regions, like motor regions, in the functional way, of course, and we try to relate them to anatomical known structures. And that's also very important because these regions bear some specific kind of almost modular, in some sense, not perfectly, of course, specificity. So, for example, face regions respond mainly to faces, also to other stimuli, but mainly to faces. Motion area, which I will talk about a bit more, respond to motion, but also to other things like disparity and depths. So in that sense, different regions are important to know and understand because they support different functional components of our complete conscious picture of the world. And that's why they're so important, that we understand them. On the other hand, it's a difference whether we talk about an area responding to faces or to motion versus saying this area reflects with certain aspects within this area different subcategorical features. For example, in the faces, we might identify a specific face, and then we might find coding principles within the area in the so-called corticolumnar patterns, which encode sub-features, for example, something about the shapes of noses, shapes of eyes and hairs and so on, in the face regions. Or in motion, it's easier, different directions of motion. That would be features within global statements that this area responds to motion. And that's exactly what you want to do by shifting the attention in neuroimaging from this macroanatomical level to the mesoscopic and partially microscopic level. And that's, of course, a great challenge because we have to bridge scales, and we need, as we see, all these levels to make some progress in this regard. We will also see that it is important to understand not only the structure within these specialized regions, but also to understand communication between brain regions. We need to understand and measure and model interactions at different laminae, which allow us, for example, to separate whether an area is driven mainly at the moment a certain task, mainly driven by input, or mainly driven by top-down. And we can only solve this puzzle with imaging by using anatomy, by sampling correctly different cortical layers as good as possible. And that's what I want to show you also. So there are two major aspects which we call usually mesoscopic and functional brain imaging. One aspect is tangentially in the cortex, so what features are represented there, and one is in depths. Can we learn something about the separation of different layers? This is just an example more for the visual cognition side of macroanatomy. So we have parceled functionally a lot of areas, these are just a few, and of course they are linked to some extent also to microanatomy, as has been worked out by many groups. Many of them are here. And now we have to, of course, zoom in, that's a bit the idea, and look what's happening inside these areas. So it's a more mesoscopic approach. And it's important that if we want to look inside, we need to know where we look inside. So if I want to make claims about the columnar organization of a brain area, I first need to find that area. And that already requires anatomical and functional information to be sure. For example, in V1, I can use at high-field imaging the Ganari stripe, which I just see there. But I can also use retitopic mapping, which helps me also to identify early visual areas, for example. MT I can localize by functional mapping of motion direction stimuli, but I can also use myelin contrast to find a spot in the lateral occipital cortex which corresponds to MT. So there are always, at least in many cases, anatomical ways, but for many areas, the only sure way or accepted way is functional. So we try to kind of integrate these different approaches. As I said, for retitopic mapping, we can use techniques like population receptor field mapping, which help us to quite precisely find the borders between V1, V2, V2, V3, and actually higher areas. And this is important because we will look in these areas in a moment, but we look then not in just, in parentheses, just the retinotopy, but we look in features inside these areas like orientation contrast, or disparity tuning, or color tuning. And this we want to map within these borders of V1, V2, and so on. I want to show you these multiple scales which we want to bridge in a very, very broad range of scales here, from the nano to the macro. Of course, brain imaging usually is more at the macro side. And when I say brain imaging, I really mean like 3 Tesla anatomy and 3 Tesla fMRI. This is mainly on the left side, on the macro side. We understand brain areas and we understand networks, how they work together to subserve certain cognitive functions. That's what we can do. And we can do this both in anatomy, in the sense of we can measure also fiber tracts, for example, but we can also do this in functional space by delineating areas using what I said before, different kinds of anatomical and functional localization tools. And now comes the important point of my talk. If we use more strong, more powerful scanning devices, we can push this a bit to the right, as shown here. So we go into the mesoscale just by having better scanners. And this, as we will see in a moment, is the way, because this little step towards the right side enables us suddenly to see things we cannot see with the usual conventional brain imaging. We suddenly are able to separate functionally different layers of the cortex and separate bottom-up from top-down processing, as I show you in a moment. We are suddenly able to see structures, columnar structures within the brain area along tangentially of the cortex. So by having ultra-high field imaging, we not get the same as the macro imaging, but we suddenly get a qualitative change. We see things we cannot see before. And that's kind of important and the motivation behind this research. Just to show you how this looks like, this is basically almost raw data. I just want to show you how this looks like with 7T in a 0.8 isotropic resolution. And you see how small the voxels become. I mean, I hope you see them. Here are very tiny voxels. Here, some are blown up so that you see the voxels here enlarged. And important is that they already are showing a functional contrast, which I will detail later, in this case of horizontal versus vertical motion. I will show this a bit more in detail later. But not in this raw form. But I wanted that you also see that if we go to 7 Tesla functional brain imaging, we suddenly get very tiny voxels and a lot of them. We also have to do statistically do proper correction for multiple comparison and so on and so on and so on. So that's kind of how this looks like. Another example is shown here. A region, the inferior colliculus, which is actually a very, very tiny region of a few millimeters, 5 to 10 millimeters, which is kind of a voxel in 3T, maybe 2 or 3, but not much, which becomes at 70 a full detailed tonotopic map. So instead of just saying there is something active when I hear something, this is auditory research, we can now even see in this voxel, so to say, of conventional imaging, a full tonotopic map where different sound frequencies are orderly projected. And that's kind of the benefit we get. We suddenly see more rich detail there. And of course, the columnar concept goes back to previous work for Mountcastle and the somatosensory cortex, but for the visual cortex, the landmark study is from Goebel and Wiesel. They got also the Nobel Prize for that. And they discovered first with electrophysiology, so by putting electrodes in the cortex of animals, cats and later monkeys, and found these kind of cells responding in visual cortex within a certain region for a certain patch of the visual field, so kind of with a constant retinotopic position. They found a different full corticocolumnar organization of the different orientations of lines in space for the same patch of the visual field, which means that the visual cortex, the primary visual cortex, acts like a filter bank, which looks not only where something is, but does more. At each position already analyzes the contours, the kind of the transitions of contours from say, darker to brighter or so. So shape analysis is starting in primary visual cortex at each retinotopic location. And that's actually shown here with optical imaging, which looks on top of the exposed cortex in animals. And you can then see in different colors, in small patches around so-called pinwheels, the different orientations forming kind of representations of these kind of lines, which are detected there by filter banks in primary visual cortex. And that's actually what we want to see with brain imaging, the human non-invasively. This is an invasive technique. That's the goal, and we show you the first steps now. Before I come to that, I want to highlight also that we touch even a bit on the micro, and that's because we are very, very lucky. If neurons would be random, so neurons coding for the same thing, like in the orientation case in V1, all the neurons responding to an oblique horizontal line, if they would be sprinkled within this so-called hypercolumn, we would not probably have a chance to see it with imaging. But we are lucky that the neurons cluster, so that the neurons with a very similar response preference in this mesoscale, they tend to co-localize. It's again an anatomical point, which gives us the chance to see this with ultra-high field imaging, because these kind of columns are supposed to be, depends on which type of columns we talk about. I talk more on the so-called functional columns. They are about 500 micron to more than a millimeter big, and these columns we will measure. And so that means if we are lucky that the kind of neurons with the same response profile cluster, for example, different orientations here in 3 different colors, then it's sufficient, so to say, to separate these columns to say something about the neuronal preferences in this area, which features are in this area if we get to this level. So that's an important point. So the lucky of nature of imaging is that kind of, at least in many areas, I cannot prove this for all, but in many areas, it has been shown in the animals, and we kind of do this now also in the human brain, that neurons with similar response profile cluster together, and this is what we want to resolve. Important is that we don't know how good this will be in depth. This is, of course, a very idealistic scenario. Columns might not penetrate perfectly well through all the layers. There might be much more complicated structures, as has been shown in the macaque monkey by Tanaka and others, for example, that it is not an idealized column. But nonetheless, we exploit this principle. And here is some proof of that this is possible by the group in Minneapolis from Camilo Goebel, with Isabelle Coup's study on the orientation columns in V1. And you see here, strikingly, that the colors here correspond resolved orientation columns in the human brain non-invasively using 7 Tesla fMRI. And you see these black lines, they correspond to the ocular dominance, which kind of is a little bit more larger clustering, which is also present in primary visual cortex for the left and the right eye. So that means we have now the possibility to look into this resolution. A problem here is, and it looks strikingly similar like what is known from the monkey using optical imaging. The problem now is that they could only do this by having a very thick slice and a very high in-plane resolution, so not isotropic voxels, as shown here. So that they basically have only one slice measured, because it takes time with spin echo sequences, which we often use next to gradient echo imaging to be more sensitive to neuronal vasculature effects and not the draining vein effect. So we use often spin echo sequences, which have a low signal to noise at low fields, but at 70 and higher, it's strong enough to get decent images with a very high neuronal specificity, so to say. And you see here, they had just one slice and they moved it in the calcarine, one bank of the calcarine sulcus, and then basically could average in a thick slice along the direction of the columns and pooled, so to say, along depths. And that was the trick that they could find this. Because if you have isotropic voxels, you would not have enough signal to noise with 500 micron in plane. We wanted to change that, so what we did is we developed tools which allow us at the same time with isotropic voxels and a 0.8 millimeter resolution to kind of measure all dimensions equally well, and we kind of conceptually separate now 3 different approaches which are in the literature on ultra-high field imaging at the moment. One is that people pool across layers. For example, you want to say, is there major input at the moment in a certain functional task, bottom up or top down in V1? Then you basically pool the amount of activity in the middle layers and separate this from upper and lower layers. That's one kind of laminar pooling. Then there is the study like we just saw from Isa Yacoub, where the pooling was vertically. So you had basically voxels which were thin here and were thick along depths here in this kind of example. And then we also have an approach, that's mainly what my group is doing. We basically try to measure both at the same time, so that we measure both tangentially columnar organization and in depth the laminar organization. That's actually the strategy we pursue in our lab. And I will show you some examples. As I mentioned before, we do not expect this kind of idealized ice cube or textbook models. We expect much more complicated structures, but we hope also to basically use imaging after showing that it really works by kind of copying results from animal research, that we can then also use this technique to also learn what's happening in human areas and also basically get new results about the organization across layers and across the cortex. And one important point is immediately apparent, that the cortex can no longer be modeled as a sheet because we need the different depths. We cannot project everything on one sheet around the cortex. We have to model depths, and that's very important. And we develop techniques to do this in an optimal way. Currently, the most approaches use the so-called mesh approach. You basically have a voxelated segmentation of white matter and gray matter and CSF, and you basically either in the middle or at white matter, you use really the voxel surface itself, then smooth it and move it up and down to basically estimate the depths and sample from different depth levels. We also use this approach, but we found that because you start with a voxelated representation and then smooth it, you get very irregularly spaced vertices in your meshes. In order to get very regular, mathematically precise distances geometry, we do it differently. We basically first smooth the intensities arbitrarily precise inside gray matter, and then reconstruct directly at any level we want new surfaces with a geometric rectangular structure we call grids. And that is a superior approach because you get very precise, theoretically precise or estimated structures without too many sampling artifacts. And we also learned again from neuroanatomy that sampling is quite tricky because we do not see, of course, in vivo, the layers of the cortex, so we have to estimate them. And we learned, talking to Professor Zillis and others, that the sampling is tricky. And actually, we were pointed out to this Bock principle, and also Werner, 2014, had a very nice paper showing how important the sampling is. And we use this now and use curvature instead of seeing the depth layer. We adjust our sampling of depths based on curvature information. And that is quite important because we see here that in kind of straight cortex, we have a certain partitioning of volume fraction across depths here. And that is not retained if we go in high curvature regions like in the sulcal fundi here or in the crowns of gyri. Here we have to kind of adjust the volume fractions based on these kind of corresponding segments, which you see here, which partition the tangential space here. And then we basically have to adjust, for example, these are much smaller than this. This region is much larger than this. So we basically have to move the layers upward here to get the same volume fraction as we get it here. And vice versa in the gyri, we have to then expand here and shrink here. So that's very important that we kind of adjust the sampling to estimate whether we are an upper, middle, or lower layers. That's the goal of our very coarse layer separation. And we do it in our grid approach because we are allowed to move and reconstruct meshes wherever we want. We basically reconstruct the meshes exactly there where we keep the volume fraction across these segments, which we fit here with this kind of pyramidal-like structures. So if we do that, we get very precise sampling. And actually, we do this, as I said, in kind of continuous space, which are the best optimal estimates of the different compartments, for example, upper, middle, lower. But then we can make out of these continuous measurements voxelated space depending on what resolution our measurements have, and then sample this to our voxel resolution, and then can partition that. And you see here the difference. You see that in the equidistance approach, where we do not adjust for curvature, the depths, we get differently looking samples as we get here, where the things are pushed out or pushed inward to accommodate for the curvature effects, the Bock principle. And how can we show and prove that this is really relevant? So we did a study with very high resolution on 9.4 Tesla, where we basically sampled V1, V2 by using population receptor field mapping, and then looked in high anatomical images with 350 micron isotropic resolution. And we basically find then very nicely, for example, you see this here in V1, the Ganari stripe. And if you look at depths and sample here, you will see this as a dip in the intensity around the middle of the depths of the cortex. And that's precisely what we see here. So this is kind of the depths of the cortex, and you see that roughly in the middle, we get this dip in intensity, which comes from the Ganari stripe, because you have a lower intensity there in these images. And now comes the interesting lesson. If we use the wrong, but we used actually for some of our work before, if you use the simple sampling, the equidistance sampling, you get a mislocalization, at least in high curvatures, cortex. You get a mislocalization that the gyra and the sulci mislocate the deepest point, which are the stippled lines here and here, too far left or too far right, so not in the middle of the cortex. Whereas if we adjust it by the Bock principle, our sampling, we get across depths always in the across the cortex, always in the middle, this dip which indicates the Ganari stripe. So that shows that indeed these anatomical principles are really relevant and valuable, and we need to use them for our high field imaging. So this is how these grids look like. I will now show you very briefly a few examples of that. So one is one of the earliest studies I did in my imaging career was motion mapping, because I'm interested in visual motion. You see that this was a very simple stimulus we used, and we could localize V5 here, the motion area, human V5, MT+. Now comes the question. We were happy at that time, and other groups as well, like Tutel and others who studied that region, because we could just see it respond stronger to any kind of motion versus static, right? But now comes what we can do at high field imaging. We can ask the question whether what features are there. So can we separate within this area regions for, say, horizontal motion, vertical motion, oblique motion, and partition then tangentially the cortex into these different features like we have seen for the orientation columns in V1. And indeed, that's possible. So we could show this with 7 Tesla, and the different colors now not show orientation, but different axes of motion, so the feature which MT is coding. And we see also that the tuning curves, which are cross-validated, we could extract from these voxels for different directions of motion. They basically follow very nicely unimodal curves as predicted from monkey research by recordings in MT. So that shows that we indeed get now to this feature level, tangential level within the cortex. And actually here you see different depths cuts through that. Roger Tutel's group, in a nice paper of Nasser recently, showed that you can also in V2 find the corresponding to the thin and the thick stripes from cytooxidase staining. You can find this also with 7 Tesla imaging back in the human brain. You can find basically these stripe patterns, which were predicted from animal research. And if you look carefully and contrast color and disparity, then you see that indeed that you get corresponding to the thin stripes, you get the color responsive regions, and corresponding to the thick stripes, roughly, you get these kind of disparity responsive regions, and they nicely complement. And that shows that we can also go beyond V1 and V2 and V3 and find these kind of columnar level organizations, or stripe-like in this case. We also did this recently for auditory cortex, just to show how general that is. And we moved in the primary auditory cortex and the regions around, and we used this grid approach and found that the primary auditory cortex has a beautiful columnar organization for tone frequencies. You get beautiful maps, that was known before, but we also could show that they penetrate in depths with the same frequency preference very nicely, but not so much in surrounding cortex. So it's a special feature of kind of textbook columns we only see in the primary auditory cortex, not in the secondary belt regions. So to summarize that part, it shows us that the columnar organization becomes in reach with imaging, but we have till now only replicated animal research. That means the future should apply these techniques, and that's of course now outlooking point, apply these techniques to areas which are not yet investigated, not even in the animal, so more specifically human regions, to learn what are the feature codes there, and do we find columnar organizations in these other regions. I want to now finally say a few words also about the laminae, because here the anatomy, as we have seen from the sampling, becomes very important. Because if we are able to separate columns and cortical layers roughly from each other, we can also separate bottom-up from top-down flow. So I want to show you two examples. One is, before ending my talk, one is for the tangential orientation and one is for the depth orientation, which give us really new insights in how the brain works. The first one is studying some conscious perceptual effects. And it's actually ambiguous stimuli. So what you see here are actually probably moving squares, maybe like that, or some might also see it like this. Actually, the stimulus is ambiguous. And actually, we show only at the corners here 2 squares, and then we show the 2 squares here. Everything else you perceive is in your brain, is in your mind. So you construct a motion either horizontal or vertical. It's a visual illusion. Right? Until now, we could not predict that because we need to find the features within MT to kind of predict what your current percept is. And precisely that is now possible. This is just a kind of physical stimulus. This is a real motion. You should now really see the real motion. And what we found is indeed, this is for horizontal versus vertical motion. In the apparent motion, you see these kind of red and green clusters in MT. This is an inside look in MT. And they code whether you see horizontal or vertical motion, if you show it physically. But now what's happening, if you see it illusionary, then we get a boost in the horizontal responding ones. When you perceive horizontal motion, subjects press a button, they say, now I see horizontal, now I see vertical. Many subjects do not even know that this is in their brain. They think the computer changes this. It's a very strong effect. And then when they press their button, we see that we get a horizontal boost of columns or a vertical one when corresponding with the percept switch in the subject's case. So we can predict the state, the conscious perception, what people perceive, whether they perceive horizontal or vertical, from high-field imaging inside this area MT. This is the novelty of this approach. We also can see using the layers, like the upper, middle, lower, top-down effects like attention. So here you see effect on upper layers, which show that the tuning for auditory information, almost done, for auditory information is much more sharp, the tuning, if you attend to auditory stimuli. And this happens only in the upper layers here. And this I will skip for time reasons. I want to show you now a last example here from my colleague Lars Mugli. He did occluded images and showed kind of either full images or occluded images. And when he showed the full images, he trained the classifier in V1 to predict what you perceive from the activity pattern in V1. Now the trick comes when you test the classifier and your brain activity. The subjects only saw these images which were 2 quadrants filled and 1 quadrant occluded. And now the classifier from the occluded region, that means from the voxels in V1, which were not stimulated, could still predict what you see, although there is no stimulus in this region. And the only explanation is that goes first from the partial image up the hierarchy in the visual cortex, then forms predictions top-down, which also include the occluded region, so they also come down there. And that's precisely what we found. We found that the upper layers, you see this here, so this is when the stimulus comes fully, then the middle layers, and actually all layers, can recognize what you see. This is shown here for MVPA accuracy. But if you have these occluded voxels, the top-down gives you this information only for the upper layers, which are here on the right side. Here's the lower layers, here are the superficial layers. So that shows again that resolving these laminae helps us a lot to resolve that. I will skip that slide because of my time is up, but I want to just summarize for you the main findings. Important point is to bridge the scales, we need to integrate macroscopic imaging, but also this new form of mesoscopic brain imaging. And if we resolve columnar-like features, then we even touch on microscopic features because neurons, which you cannot measure directly, cluster. So we get some hint on the coding principles in the brain. The laminar resolution helps us to understand effective connectivity because we have not the temporal resolution in fMRI, but if we can see what is mainly coming in and what is mainly coming top down, we start to be able to make arrows on our information flow directions. And I think these kind of two mesoscopic approaches will help us to unravel more in the future how the human brain works. Thank you very much.
So do you think that these methods are actually feasible in clinical populations that tend to move more than controls?
That's a very good question. If the voxels become sub-millimeter and you move 3-4 millimeters or even more in patients, it would be useless. So therefore, it's very critical, and we do a lot of effort in that to ensure that we have basically sub-millimeter motion. You can do this in two ways. One is good post-processing, but the best is better recording. So, of course, then you need compliant subjects, but you need also so-called motion cameras, which track the scanner. And Benedict Pose in our lab has implemented that for our sequences so that we basically use this more and more in the future to basically get not the motion in our images in at the first place. If you then average over time, you see details like tiny vessels because it proves that you really have now aligned the images over time perfectly well. Now to the patient groups. This is not yet much done in patient group. We have a 7 Tesla study on neurofeedback using this kind of work with Heidi Johansenberg, but nonetheless, it's more difficult, and we throw out a lot of runs which have too much motion, I have to be honest. So we cannot not use it now routinely in clinical populations, but we work hard also to use motion cameras and so on, so to get, which were made for that, for clinical populations originally, to get really this also into clinical. For example, in Liège in Belgium, they also get for clinical population a 7 Tesla scanner soon, Stephen Laurie's group. Also in Glasgow, they got a 7 Tesla for clinical work. So that means we have to solve these problems, and it will be tough, but I'm sure it will come good clinical applications in the near future.
And how do like cell recording action potential come into this picture then?
Of course, this is a very good but challenging question to answer shortly. I mean, this is indirect. So if you use fMRI, you measure somehow input in brain area. So it's closely related to LFP, as has been shown by Lokothetis, who recorded simultaneously in the same region spiking, local field potential, and the fMRI, the bold signal, and found that the spikes are too local and to correlate not very well. But the little bit more spatially integrating local field potential had a quite high correlation with what we measure here. So you measure not spikes, but you measure more kind of integrative signal, which reflects this kind of mesoscopic scale population responses, like a local field potential.
Okay, thanks.